Okay, my name is Jessamine Burke, and I'm going to be doing a video on the complement system. The complement system is one of our body's main defensive mechanisms against different pathogens, and it's basically just proteins that are circulating our bloodstream continually, and they're inactivated, and they can become activated in response to a pathogen evasion. Complement functions in both innate and adaptive ways. Alright, so let's just get into. Alright, there's three major pathways. The first one we're going to talk about is the classical pathway, and it's initiated by antibody-antigen complexes. And so this pathway is part of the adaptive immune response. And what that means is that a person would have to have had prior exposure to that pathogen since they have produced antibodies against it already. So the classical pathway consists of a C1 complex, which is basically just a protein complex that's composed of three different subunits. There's 6 molecules of C1q, 2 molecules of C1s, and 2 molecules of C1r. And for it to become activated, the constant region of either an IgM or IgG must bind to to the C1q molecules. And whenever that antibody binds to the molecules, it produces a conformational change, which will also result in the activation of the C1r and the C1s. And the C1s is actually a serine protease, and that just means that it'll go on and activate other proteins by cleaving them. So the C1s will activate the C2 and C4 complexes and cause them to be cleaved into the C2a and C2b, and the C4 into the C4a and C4b. And the C4b and the C2a subunits will combine to form the C4b2a, which has catalytic activity, and it's known as the C3 convertase. And what it does is it'll cleave C3 molecules. It'll cleave it into C3a and C3b. The C3a is just an anaphylaxitoxin, and so that just means that it'll float off and cause histamine release from other cells, which will just increase the inflammation present. But the C3b is the more important subunit, and it can function as an opsonin, which means it'll just increase phagocytic activity. Or if it's produced in excess amount, it can bind to the C4b2a, and it will create the C4b2a3b, which is known as the C5 convertase. And the C5 convertase is a much more stable molecule than the C3 convertase, and the C5 convertase will cleave the C5 component into C5a and C5b. And once again, the C5a is an anaphylaxitoxin, and the C5b is the portion that will form the membrane attack complex. It'll associate with the C6, which will activate the C7 and then the C8 and the C9, and they all come together to form this complex that will actually punch holes into the membrane of the invading pathogen cell and eventually cause it to die. Okay, so the second pathway is the lectin pathway, and the lectin pathway is part of the innate immune response. It doesn't require the presence of antibodies, so it can be activated without prior exposure to a pathogen. And it's activated by the presence of an unusual sugar, molecule that's usually found on lots of bacterial cells. So the pathway uses lectins to recognize these molecules, and the first lectin that was found that could initiate the activation was the mannose binding lectin, or MBL for short. And in the serum, MBL is associated with a MASP protein, and MASP is just a protease that will cause the fragmentation of C2 and C4. And so then, this pathway will basically follow the same as the classical. Really the only difference is that it doesn't use the C1 complex. The lectin receptor is basically takes the place of the C1 complex, because it's, I guess you could say, the antigen recognition receptor, and the MASP functions as the C1s does, because it's a protease. So the lectin and the classical mechanisms are very similar, but really the biggest difference is that the lectin pathway is from the innate immune system, so you don't have to have had prior exposure with the pathogen, as with the classical pathway, which you do. Okay, the alternative pathway is also part of the innate immune response, so it doesn't require the presence of an antibody. It is actually activated due to C3, because C3 is very reactive, and it will spontaneously fragment. So it'll fragment into the C3A and C3B. And then the C3B component will bind to just another protein, which is called factor B, and then factor B is cleaved by another protein known as factor D. It'll cleave factor B into big B little a and big B little b. Big B little a is the smaller subunit, and so it'll float off. But the big B little b is the catalytic part, and when it's bound to the C3B, it forms C3B, big B little b, which is known as the C3 convertase. And so the C3 convertase will cleave more C3 molecules, and so which and the C3B portion can do the same thing again, and can bind back to the C3 convertase, and it'll form the C3 little b, big B little b, big B little b, which is the C5 convertase. And then the C5 convertase forms the membrane attack complex, just the same with the other pathways. But the alternative pathway is different, though, because it's just, it's spontaneous, because C3 is always fragmenting. But under normal conditions, like when we don't have a pathogen in our bodies, our normal cells have an inhibitory protein, which can deactivate the C3b molecule if it tries to bind. But bacterial cells don't have an inhibitory protein, so when C3b bumps into those cells, it'll bind and activate the alternative pathway. Yeah, that's labeled wrong. Sorry about that. Anyways, so yeah, with bacterial cells, it'll bind to, and the alternative pathway will take over. And also the C3b is more attracted to cells with a slight negative charge, So that also causes them to be more attracted to bacterial cells as opposed to our cells. Let's see. Yeah, this is all, that heading's wrong. That's supposed to be the alternative pathway, but that just shows you the steps that we've already been over. And here is an overview of all three of the pathways. The pathways all function, the same way. Their main goal is to get rid of the pathogen, but we have evolved to accomplishing this in different ways, because as bacteria and viruses mutate and develop different mechanisms, we have to do the same.